Hi students, once again I am meeting you to discuss about the chapter that is Find, Digestion and Absorption. In the previous classes we have discussed about the various events of digestion. Now we have to pass on to the next one, absorption process. Now as a result of digestion we have received monomeric units or symbol units like glucose, fructose, galactose, amino acids, fatty acids and glycerol. And all these substances enter into the small intestine and they are being absorbed in the ileum and jejunum region. The unabsorbed, undigested materials are propelled into the large intestine. Now the various activities of the digestive system are under nervous and hormonal control for the coordination of the different organs. For example, the gastric juice as well as intestinal juice secretions are normally stimulated by the nervous control. That is, the presence of food stimulates the gastric juice, like that. And the hormonal control of digestive juice is normally initiated by the local hormones or stimulated by the local hormones. The meaning for local hormones released by the wall of the intestine itself, that is called local hormone. So the digestive juice, the hormonal control of digestive juice, normally stimulated by the local hormones of gastric and intestinal mucosa. This is a simple regulation. Now I mentioned about the absorption and then assimilation. Let us take first the absorption of the different materials like carbohydrates, proteins and fats. First of all, what is absorption? We have small intestine. The wall of the small intestine, you know that one, having intestinal mucosa. If you are taking, for example, the lumen of the intestine is symbol 1. So this is the wall of the intestine. We have the simple substances formed in the lumen of the intestine. What is absorption? So it is a process by which, you see that one, the end product of digestion. The end product of digestion, nothing but the glucose, fructose, galactose and also amino acids, fatty acid, glycerol, etc. of digestion passes through the intestinal mucosa. Now this is the intestinal mucosa. The wall of the intestine, the innermost layer is called intestinal mucosa lined with epithelium. So the product of digestion passes through the intestinal mucosa into the blood or into the liver. That process is called absorption. This is a simple definition. Okay, that is what is called absorption. It's a process by which the end products of digestion passes through what we have actually the inner layer called mucosa layer lined with epithelium. Enter either into the bloodstream or into the liver. Now we see that one in the iliac region we have already studied the unit of absorption. The unit of absorption is nothing but what we have the villi region. Now the villi in the lumen of ileum are considered as the absorb unit, considered as the absorbing units. We studied already consisting of a single layer of epithelium enclosing a middle lacteal vessel surrounded by a fine network of blood capillaries, both arteries and veins. Normally there is a structure of villus we have studied already. Okay, so it is bound by a single layer of epithelium enclosing a middle lacteal vessel, nothing but the lymphatic vessel, surrounded by a fine network of capillaries. Now what is the mechanism of absorption? The absorption of materials of different types is not uniform. Different materials are absorbed in a different way. As per the syllabus, 3 methods are given. What are the different methods of absorption? Number 1, passive transport. Number 2, active transport. Number 3, facilitated transport. Passive transport, active transport and facilitated transport. We will see one by one what is passive transport, what is active transport and also facilitated transport. Now the first one, passive transport. What is passive transport? Otherwise it is called downhill movement. That is, the molecules are moving from an area of higher concentration to an area of lower concentration, higher to lower, without spending energy. Downhill movement, without spending energy. So, it's a simple diffusion process. You see, and the passage of these substances depends on the concentration gradient. Concentration gradient means concentration differences. Suppose let's take see, I mentioned about the passive transport, 3 substances, some glucose, some amino acids, and some and chloride ions, not some. So we have the electrolytes of chloride ions, some glucose molecules, not all, and some amino acids are transported by passive transport. Now let's assume, for example, if you have chloride ions here 100 in the lumen of the intestine. Now this is the lumen of the intestine, this is a wall, what's called the mucosal layer. Now here we have the blood. Suppose we are having the blood stream. This is the area of blood. Now what's happening? Now the chloride ions are nearly 100 molecules, or 100 just we have, that is units. Now in the blood, say an example, we have only 60 units. Now without anybody's help, what will happen? From higher concentration to lower concentration, the molecules are moving. That's called downhill movement. No spending of energy. So what are the molecules normally transported by this way? The electrolytes, chloride ions, then some glucose molecules, not all, and some amino acids. So higher to lower level. Now the next one, see, active transport. The transport of materials against concentration gradient. In passive transport, along with concentration gradient, that is along with the meaning from higher to lower. But in the case of active transport, the transport of materials normally occurs against concentration gradient. For example, we are having just actually glucose, amino acids, sodium ions. Major amount of glucose, major amount of amino acids, and completely sodium ions are transported by active transport. Let us assume I am taking, for example, the glucose molecules here just normally just 100 and here 80. So the number of molecules of glucose 108. What will happen normally in a normal process? The molecules are moving from higher to lower concentration. Okay, this is one. I'll take another example, just a simple way. Here let's assume there are 100 molecules, here less number. So higher to lower, that's called the passive transport. Now I am taking just 100 glucose molecules in the blood and 80 only in the liver. What will happen normally? The movement occurs from, that is the blood, into the liver, but it doesn't occur. Now we need more and more glucose to release energy. That's why what will happen? The cells pick up actually the glucose molecules, though they are lower in concentration in the blood, by spending energy, capturing the molecules and transporting the molecules into the blood. For that, we have to spend energy. So, that's why I say, the transport of materials against concentration gradient. Against means from lower to higher. Here, along with, from higher to lower. Passive transport, along with, from higher to lower. But here we see that one, from lower to higher level. It is being normally picked, it's not moving. This is called upstream transport or uphill transport. This is downhill transport, passively they are moving down. But here it is called uphill movement because we need energy for climbing what is called a mountain. Likewise here, the molecules at low concentration are moving to a molecule of higher concentration. For that we need to spend energy. So that's why I say the transport of materials against concentration gradient by spending energy is called active transport. So otherwise called as what is called uphill transport. Uphill transport. Uphill. This is what is called downhill transport. Downhill transport. So these are the two major ways of transport. Now another one what we call facilitated active transport. Facilitated active transport. Here the molecules are moving on their own, whether with the help of energy or without the help of energy. But what do you mean by facilitated active transport? The transport of materials with the help of a carrier molecule. So here no carrier molecules are required to carry these molecules into the blood. But here, in the case of facilitated transport, what I mentioned, F refers to the fructose. So the fructose need a helper molecule, a carrier molecule for its transport. They cannot move on their own. So the transport of materials with the help of a carrier molecule is called facilitated active transport. Here I mentioned F, that is nothing but fructose. So G for glucose, A amino acid, this is sodium ions, likewise chloride ions, glucose amino acids. Okay, so the transport of materials with the help of carrier molecule is called facilitated transport or facilitated active transport. For example, the transport of fructose by sodium ions. Okay, that is the third type. And the other types we have to see the absorption of different materials on their own or by means of different mechanisms. We'll discuss one by one. I already explained what is facilitated transport. The transport of materials with the help of carrier molecules. Example, transport of fructose with the help of sodium ions. Now, the materials are absorbed either actively or passively. But about the fatty acids and glycerol or absorption of fat, it requires a different mechanism. Now, absorption of fats. What is happening? You see the fat-soluble vitamins A, D, E and K. Then we have the glycerol, the fatty acids, and then the monoglycerides, not mentioned in the book, monoglycerides, made up of one molecule of fatty acid, one molecule of what we have glycerol. All before absorption incorporated into a small spherical water-soluble droplets. These small spherical water-soluble droplets are called micelles. See, many micelles are formed. Now this is the micelles. The red one indicates a central core of what we call the fatty acids plus monoglycerides, even cholesterol also, which is being surrounded by the bile salts. So this is what's called micelles. A micelles consists of a central core of the fatty substances surrounded by what is called bile salts. Now the micelles are nothing but what we have the droplets, water-soluble droplets formed after mixing with the bile salts. Now what will happen to these bile salts? We have to go further. The absorption of micelles, how it happens, etc. So these droplets are called micelles. The short answer. So what are micelles? This is the answer for that one. Except fat, all other materials are absorbed directly into the bloodstream. So what is happening during the absorption of fats? They are first incorporated into micelles. Now these micelles are acted upon by the pancreatic lipids. Now what I represented here is nothing but the lumen of the intestine. This is the wall of the intestine. Then I represented what we have, a lacteal in green color and a blood vessel. A simple diagrammatic one. Now the large piece of fat enters into the small intestine lumen. So mixing with what is called the bile salts, we are getting what is called water-soluble micelles. The micelles are acted upon by what is called the enzyme, the fat-splitting enzyme, pancreatic lipase. So that we are getting the fatty acids and then monoglycerides. Now the fatty acids and monoglycerides are transported into the epithelium, the lining of the lumen of the intestine, by means of pinocytosis. Pinocytosis means cell draining. So they are being taken into the mucosa, the epithelial cell, where they are once again resynthesized inside the endoplasmic reticulum to form fat globules, micelles, fat droplets. Here after resynthesis, there is nothing but we have the union of the fatty acids and monoglycerides to form just what we have the fat globules. These fat globules are called chylomicrons. The chylomicrons are synthesized in the endoplasmic reticulum. They are moving through what is called the cell of mucosa by means of exocytosis. And the invagination is being formed, and then what we call endocytosis means taking in of what is called any material by the cell. Here exocytosis means giving out the material by the cell. So by means of exocytosis, Now the chylomicrons enter into the lymphatic duct, what is called the lacteal present within the villi. Okay, so this is what is happening. So reason is from the chylomicrons, they enter in the lacteal, simply I am telling that one, within intestinal villi, and subsequently empty into the lymphatic duct. So from the lacteal of the villi, they are all released into what we call the lymphatic duct. Now all these lymphatic duct join together to form a large lymphatic duct. So what will happen? It receives the lacteal vessels of different villi join together to form a large lymphatic duct. Now the absorbed substances by the lymphatic duct are released into the bloodstream. So ultimately the fatty acid glycerol enter only to the bloodstream. Though the absorption is by the lacteal vessels, but the circulation of these fatty substances to different parts of the body for assimilation is only through the bloodstream. So by exocytosis, the chylomicrons enter into the lacteal, then into the lymphatic duct consequently. Then what is happening? The absorbed substances by the lymphatic duct finally enter into the bloodstream. So, while fatty acids and glycerol, these are all absorbed by the lacteals, whereas other materials like glucose, fructose, amino acids or electrolytes, they are being absorbed either actively or passively by the blood directly. So, fatty acids, glycerol are absorbed by what we call the lactate, the lymphatic duct, whereas other materials like glucose, fructose, galactose or amino acids or electrolytes, they are being absorbed directly to the bloodstream either passively or actively. You know that one, the water-soluble vitamins. These water-soluble vitamins are absorbed. For example, vitamins B and C. And we see the fat-soluble vitamins are absorbed through the lacteals, whereas the water-soluble vitamins B and Z are absorbed just either by means of simple diffusion or by means of active transport. Either by means of simple division or by means of active transport. The absorption of water depends on osmotic gradient. Water is moving from an area of what is called lower concentration to its higher concentration. That is what is called, normally I can say it's in a reverse order. We have different definitions for osmosis. So the movement of water depends on osmotic gradient. It's moving from what is called actually lower concentrated solution to higher concentrated solution. So according to the need of requirement of water, water is moving from the blood or from the intestine into the blood, or simply there is no absorption. That depends on the amount of water available in the blood. If the amount of water available in the blood is less, more water is absorbed. If the available water in the blood is more, there is no absorption of water. That's why absorption of water depends on the osmotic gradient of water in the blood. Depends on water concentration. Okay, so that's about what we have the absorption of fat and other materials. Now we have to go for the absorption of materials in different places. What are the materials which are being absorbed at different places? Let's see now. Now, I already mentioned about what we have, the absorption of fats. Now I am writing on the board what are the facts I have described so far. Now the substances absorbed by the lymphatic duct are then released into the bloodstream. Now we have the fatty acids and glycerol while absorbed by the lacteals. All other materials are absorbed either passively or actively into the bloodstream. I explain about it. Water-soluble vitamins are absorbed either by simple diffusion or by means of active transport. Now the absorption of water depends on osmotic gradient. More the water in the blood, water absorption is less. Less the water in the blood, absorption of water is more. That is what I mentioned about earlier. Now I represent a simple list in how far actually the course of fat absorption occurs. And then we have to go for what is called few lines about assimilation process. Here I represented a simple equation showing the absorption of fats. I represented the diagram, now simple illustration. The large fat pieces normally mix with the bile salts to form what we have the small soluble droplets micelles. The micelles are acted upon by the pancreatic lipids. Then they are being converted into monoglycerides and fatty acids. They enter into the intestinal mucosa, where they are being resynthesized to form fat globule called chylomicrons. The chylomicrons normally by exocytosis from the epithelial cells enter into the lymphatic duct, namely the lacteals. The lacteals join together from different villi to form what's a large lymphatic duct, which ultimately enters into the bloodstream. Okay, this is one what I described earlier. Now we have the absorption of substances taking place in different parts of the alimentary canal. Mouth, that what we have the stomach, small intestine and large intestine. These are all the places of absorption of different materials. If you are taking mouth, some drugs are absorbed by the blood capillaries. Those blood capillaries lying below the tongue. The drugs are also absorbed by what we have the mucosa of mouth, the lining layer. That's what only drugs are absorbed. Now in the stomach, we have the simple sugars, alcohol and some medicines are absorbed. What we are taking in? Simple sugars, alcohol and some medicines. Then small intestine, this is a major part of absorption taking place. Major absorption of the substances along with water normally taking place in the small intestine. Glucose, fructose, galactose, amino acids, fatty acids, glycerol, etc. That's why I didn't mention that one. And what are the materials that are absorbed in the large intestine? More amount of water. Then vitamins. Some minerals and certain drugs are absorbed here. Some minerals and drugs are absorbed here. So that is about the absorption process. Associated with this absorption we have what's called assimilation. I will give you a paragraph. What is assimilation? A few sentences. Now what is assimilation? We are what we eat. Any food that is being taken into the body is converted into body substance. That process is called assimilation. That's why I mentioned here also, we are what we eat. Whatever food we are taking in are being converted into a body substance, namely protoplasm. Now the absorbed food materials are transported through blood and lymph to the liver, through a portal system or a portal vein, what is called a hepatic portal vein or hepatic portal system. I will talk about what is meant by a portal system and a circulatory system, a separate topic. What is portal system, what are the types. Okay, and from the liver, the absorbed food materials are transported to all other regions of the body for utilization. Okay, normally the body tissues utilize the absorbed materials for their various activities, and also they incorporate these materials into their protoplasm. Incorporate these materials into protoplasm, nothing but actually the conversion of the food materials into body substance or protoplasm of the cell. This process is called assimilation. Simply we can say the conversion of food into body substances. Conversion of food into body substances is called assimilation. Now I want to conclude with one question. So what features of the small intestine that enable it to absorb digested food effectively? This is a question there in the book box. You have to answer it. So I will give the answer for that one. So normally it is a small intestine which absorbs the various materials effectively. What's the answer for that one? In brief, I want to give just 3 or 4 points only and write it down on the board. I asked one question. What are the factors that enable the small intestine to absorb the various materials effectively? The reasons are: number 1, you know that the small intestine is the longest part of the alimentary canal, so which provides a larger area for absorption, nearly about 6.25 meters, longest part. The second one, the inner mucosa has produced into permanent muscular folds. I asked the question also in 2 marks. The inner mucosa has permanent muscular folds called wall of Kerkring. Some mistake in a book, so this is the correct spelling, wall of Kerkring, or plica circularis. This is also, we can take that one for 2 marks question. What is valve cream? You can answer that one. Even you can use that one for your public examination. Inner mucosa has permanent muscular folds called valve cream or plica circularis, which increases further the surface area of absorption. The third one, you know that one, the inner mucosal layer of the small intestine is raised into, particularly the ileum region, 4 million finger-like projections called villi. So presence of villi, another factor to make the small intestine absorb effectively. Now the cell's lining, what we have the villi, each epithelial cell has hair-like projections called microvilli, which further increase the surface area of absorption. So because of these adaptations, the surface area of small intestine increases about 60 times, which make the absorption process effectively. So the process of absorption is very effective in small intestine because of reasons 1, 2, 3 and 4. Maybe asked in 5 marks question, the question is there in your book in the box. So follow this one. And the point number 2, just we have 2 marks question. So whether I concluded what we have absorption and assimilation, we have to go further, the digestive system, the digestion process, the calorie values, etc. in the next class. Okay, thank you. I'll give the assignment and do it